So our last learning unit here is just a single lecture that we're going to have on nucleic acids. And so this kind of wraps up then our discussion of the last three learning units, which were on our biomolecules. So they were on lipids, they were on proteins, and now on nucleic acids. So we were very carbohydrate focused through the first half of Biochem 2, kind of dealing with learning metabolic pathways in the context of glucose and carbohydrates. And then we shifted to study lipids as another fuel molecule, proteins not necessarily as a fuel molecule, although it can be broken down for fuel. And then we're finally now with nucleic acids, which, yes, they're a polymer, but we don't really consume them from a fuel standpoint. Nucleic acids really, again, are all involved in information, so an information monomer. And really interesting to think about is every food source that you consume, because food was once living, has its own DNA. But again, we're not going to use that DNA that's there as a building block or as a fuel even really in and of itself. So the focus that we're going to be thinking about is really how we think about using those as a necessary building block for proper cell growth and division. And to that end, when we think about sort of applications of this, we're going to be talking about how it relates to chemotherapy agents. So again, conceptually, we're going to begin in the beginning here with just a review of our four biomolecules that we've talked about thus far: carbohydrates, lipids, proteins with amino acids, and now nucleic acids. And so good time to sort of review some fundamentals that we have there. And then again, we'll talk about catabolic and anabolic pathways for both the purine and pyrimidine nucleotides, talking about how they're related, recipes that we use for sort of building these molecules, and some important intermediates here. And again, wrapping up with two sort of disease or application-based components, talking about chemotherapy agents, and then the disease gout, which is a malfunction in uric acid production and excretion. All right, so again, first slide here, just a review, kind of important concepts to think about. Again, two main biopolymers here that are necessary as a fuel. And then these other two here really are important as information and building blocks. So again, as we learned in our last learning unit, proteins broken down to amino acids can either be rebuilt into other proteins. If they're in excess, they can be recycled. But amino acids are also really important for building a lot of other biochemical signaling molecules. Didn't focus on a lot of them, but we addressed a few in that learning unit. Again, carbohydrates really are nature's favorite hydrophilic circulating fuel. Triglycerides are nature's favorite storage fuel, again, largely because of their large caloric value. And so we spent a lot of time kind of comparing and contrasting these different biological molecules. So now as we sort of get into this learning unit, don't ever forget, right, how to draw sort of a basic phosphodiester backbone. But what we're really going to be dealing with is more kind of the monomers and these building blocks that we have here. So again, we'll address this in our next slide a little bit here, but when we have nucleotides, they are polymers made, or when we have nucleic acids, they're really polymers of nucleotides. And nucleotides, that T reminds us that there's three things that are there. Nucleotides exist of a base, a sugar, and between 1 and 3 phosphates. So again, some important things to just highlight here with nucleic acids and nucleotides is they are not designed to be a fuel molecule. We don't have a caloric value that we assign to them. They are not essential. We can synthesize everything that we need, so we do not actually need to consume anything in terms of we don't consume DNA in order to break it down and make our own. But we do have to think about sort of endogenous and exogenous sources, right? All food has DNA, but as our cells turn over, we have our own cellular DNA that we need to think about. So from an overview standpoint, right, we kind of have this concept that this top part really talks about how we synthesize nucleotides. And so this is sort of a synthesis process. So we're talking about anabolic pathways here to synthesize purine or pyrimidine nucleotides, remembering that purines are A's and G's, pyrimidines are C's, T's, and U's. And then we've got in this bottom half here sort of the breakdown of these nucleotides, or the catabolism of them. So again, we're going to kind of deal with each one of these a little bit separately, but this is a nice slide to kind of overview things here. So if we've got nucleotides, remember nucleotides are a base with a sugar, and between 1 to 3 phosphates. So if we're just talking about that nitrogenous base, we'd call it a nucleobase. Base plus sugar is a nucleoside, and then base plus sugar plus phosphates is a nucleotide. So as we learn in the next couple of slides about how we build these, we actually build purine and pyrimidine nucleotides a little bit differently. But at the end of the day, they all involve having a sugar. We're going to have phosphates that we're going to be adding from ATP via kinase chemistry. And then we build the nitrogenous bases, generally from glutamine, aspartate, and glycine. So some providing sort of carbon atoms, some providing nitrogen atoms. So one of the things that's going to be important as we think about the degradation processes is if we've got nucleotides, we need to think about snipping off the phosphates, and that's really just phosphatases that are going to be able to kind of handle that. But then we've got a sugar that we need to snip off, and once we get to ribose, then we have no problem sort of taking care of that in terms of it being a carbohydrate. But then purine nucleotides are going to be broken down into uric acid, and we'll talk about why that's problematic from a disease standpoint. Pyrimidine nucleotides, no problem sort of breaking those down into citric acid cycle intermediates and malonyl-CoA. So a couple of other things to just kind of highlight on this overview or summary slide is when we're talking about ribonucleotide triphosphates and then deoxyribonucleotide triphosphates, we're going to see this again on another slide here, but how your body works to synthesize these is it first synthesizes the riboflavor of nucleotide triphosphates and then uses an important enzyme that will convert ribonucleotide triphosphates into the deoxy flavor, and this enzyme is called ribonucleotide reductase. So RNR is really important for kind of doing some important biochemistry here that we'll talk about. So just a little bit here when we talk about sort of the synthesis of purine and pyrimidine nucleotides. So we're going to be focusing kind of on what we highlighted up here. Some important distinctions that I want you to make. Obviously, I want you to know the differences between which nucleotides are purines and which are pyrimidines. But when we talk about purine synthesis, what we do is we build the base on the sugar. And in contrast with pyrimidines, we build the base first and then we attach the sugar. So if you think about purines beginning with that ribose sort of scaffold, and then you build the nitrogen and carbon atoms in that two-ring system on top of that sugar. What happens to build A and G is you actually, again, take that ribose 5-phosphate, you build the base on top of it, and you build this common intermediate base that we call inosine monophosphate, or IMP. So this is kind of a common intermediate that we can do further modification to make it into A, or different modification to make it into G. And then we're going to have kinase activity that's involved in adding the second and then the third phosphate here. So again, purines, just to summarize, we build the base on the sugar, we get to a common intermediate that is related to both A and G, and so then we sort of bifurcate this pathway to synthesize either A or G. What's important about this pathway is whether we've got AMP or GMP or the ADP or GTP flavors of this, all of these feed back to inhibit this synthesis. Again, if we've got plenty of these nucleotides around, we don't need to continue to manufacture them. So let's talk a little bit about pyrimidines here. Again, we're going to build the base first, and then we're going to attach it to a sugar. So I do want you to know what the base is built from. So again, a common carbon source when we're building a lot of these molecules is bicarbonate. Okay, then we need some energy, so we're going to have ATP, and then we're going to use glutamine. We're going to see a lot in our next couple of lectures here when we move into integrative metabolism, talking about the importance of glutamine as kind of a building block. So we're going to use glutamine and bicarbonate with some energy to build the base, and then we're going to attach it to the sugar. So we're going to get to this common intermediate here that's called carbamoyl phosphate. And so this should say phosphate. And so carbamoyl phosphate is going to be this intermediate that we then add on to our sugar. So again, we built this base first, and then we add it on to our sugar, and then we actually get U. We get UMP. Some kinase activity gets us up to UTP, and then we do a little bit of modification to convert U into C. We didn't have to learn the structures of our nucleotides other than adenine, and so we probably can't visualize kind of the changes that would happen here, but pretty basic changes to change U into C. And then what we have back is we have a feedback inhibition loop. When we've got lots of C's that have been made, we feed back and we inhibit this enzyme called ATCase. I used to spend some time in Chem 331 sort of talking about this as an enzyme, but we really didn't focus on it in our focus on in Biochem 1. But this again is the enzyme responsible for synthesizing this carbamoyl phosphate. That again is this base that we build first, then we attach it to a sugar, do some further modification to get it into UTP, and then additional modification to CTP. One of the things that we're going to then focus on in the last little bit of this chapter is this chemotherapy agent. So obviously the building block T is going to be important for nucleotide or for nucleic acid synthesis because we build DNA from A, G, T, and C. And so we need to have a way of building this DNA building block. And so what we actually do, and we'll talk about this process, is we take UMP, so this is the riboflavor, ribo-UMP. A ribonucleotide reductase is going to convert it into the deoxy flavor. So we actually have a deoxy flavor with a U base that's on there. And I do want you to know that the difference structurally between U and T is a single methyl group. So adding on a methyl group is going to be really important chemistry. And we're going to talk about things like MTHFR again because that sort of methyl transfer chemistry is really important to this process. So one last thing to sort of add on here. Again, if we had spent more time studying the enzyme ATCase, we would have discussed the fact that A and G sort of feeds forward and activates ATCase. And this makes sense because if you're making lots of A's and G's, you better make the C's and the T's that are going to be used as their base pairing partners. So that's why A and G feed forward activates ATCase to make more of its base pairing partners, C and T. All right, so we really didn't spend too much time on this slide. I don't need you to know the details of it, but just kind of highlighting that when we make our purine nucleotides, we're going to make this common inosine intermediate. And again, we're going to build the base on the sugar. So we start with that ribose, and then we're going to build our base on top of it. And so when we have our base, we're going to again get some nitrogen atoms from amino acids, we're going to get some carbon atoms from bicarbonate, and we're actually going to get some carbon atoms from formate. So you do not need to know the details for this, but it's kind of interesting to see all the different sort of ingredients, if you will, and many of them are amino acids that we use to build lots of things in our body. Same thing here when we talk about pyrimidines. So again, remember for pyrimidines, we build the base first, and then we're going to attach the sugar. So we're going to have lots of chemistry that essentially builds this single ring. We have the double ring for purines. So for pyrimidines, we have the single ring here. We're going to, again, build that first and then attach to the sugar. And we're actually going to build U first. And then from U, we're going to convert to C and then T. All right, so a lot on this slide here. Several homework questions were addressed here. So let me start down here first with talking about homework question 4. I just wanted to reintroduce and have you remember what the central dogma is for biochemistry, that DNA is transcribed into RNA, which is translated into protein, right? And why I brought this up is we normally have this thought process of DNA to RNA to protein. But what we see here is when we do nucleotide synthesis, we actually make the RNA flavor first, and then we have ribonucleotide reductases that convert that into DNA nucleotides. And the reason we do this is there's actually way more ribonucleotides in your body than you have deoxyribonucleotides. The only real thing that you use deoxyribonucleotides for is for synthesizing DNA. Nucleotide triphosphates are used for RNA. We've got lots of RNA in our body, whether it's messenger RNA, transfer RNA, ribosomal RNA, or again, just thinking about a lot of the nucleotide derivatives that we have in our body are generally ribose flavor. All right, so question 5 just asked you to sort of think about drawing structures for things. So again, that's just what's shown up here. When we have a nucleotide diphosphate here, that's a ribonucleotide diphosphate. Again, you could put any base that you'd have on here. The enzyme ribonucleotide reductase is what's responsible for doing that chemistry that converts that 2' hydroxyl into a 2' just hydrogen there, so making the deoxy flavor that we have there. So thinking a little bit more about these ribonucleotide reductases, again, here's our 4 different ribonucleotides, A, G, C, and U. When we make deoxyribonucleotides, what happens is that ribonucleotide reductase chemistry converts all 4 of the riboflavors into the deoxyriboflavors, which means we have deoxy-UMP there. Now you'll note this guy isn't boxed here because when we're talking about the used deoxyribonucleotides, it's A, G, C, and T. So this is sort of an intermediate that we have there. We make the deoxy-U, and then that is methylated to convert that into the T. And this methylation chemistry is the chemistry that we're going to focus on in a little bit more detail here. So that's what's shown in this panel that we have up top here. All right, so we've got our deoxy-UMP. So that's what we have right here. And we've got this THF, this methylene tetrahydrofolate. We're just going to call that THF. THF is an important methyl transfer agent. Lots of things are going to get methylated in your body, and THF happens to be one of a very important set of methylating agents. So THF is going to transfer its methyl group to UMP, and you can see it adds right at this C5 position. That's how we make DTMP. Okay, so again, we've got the deoxy version that was made by ribonucleotide reductase. Then we're going to add a methyl group on to the base, and that changes that into T. The enzyme responsible for doing that is thymidylate synthase. So we're going to see that on our next slide because that is a very potent target for chemotherapy agents. One of the things that we need to think about, though, is after THF has sort of done its chemistry, it becomes DHF. So it's lost its methylating agent. And so what we need to have is an enzyme called DHFR, dihydrofolate reductase. So it takes dihydrofolate and it reduces it and it makes it back into THF. So it adds that methyl group back in, so it's ready to do some additional methylating chemistry. So 2 enzymes that we're going to talk about on the next slide have to do with the actual enzyme responsible for synthesizing T, which is thymidylate synthase, or DHFR, which is an important enzyme responsible for regenerating the important cofactor that's needed by thymidylate synthase. All right, so again, here we've got this chemotherapy sort of angle to think about. So again, the important chemistry is you need to make T as a nucleotide if you want to be doing DNA synthesis. So again, hallmark of cancer cells, they grow and rapidly divide, so they're undergoing DNA synthesis very rapidly in order to make and divide those cells. So you need to have T for that. So if you find a way to inhibit thymidylate synthase, you directly inhibit the synthesis of T. And if you inhibit the synthesis of T, then you inhibit DNA synthesis. And we've talked about this already, but a hallmark sort of cancer cells is they are rapidly dividing all the time. Most healthy cells undergo a senescent period where they're not rapidly dividing. So chemotherapy agents, again, are going to be things that don't affect most of your healthy cells. They can, because they are more than likely than not in sort of one of these senescent sort of phases where they're not actively growing and dividing. But again, we've mentioned this before, cells that are rapidly growing and dividing are things like your hair follicles, things like the skin cells in your gastrointestinal tract, and that's why you see a lot of the symptoms that you see, or the side effects, if you will, of chemotherapy agents, because they do affect those rapidly dividing cells as well. All right, so we're going to talk about this guy for a second here. This is a fluoridated analog, so a fluoro version of DUMP. This is what we call suicide inhibitor of thymidylate synthase. So this is so strong of a competitive inhibitor, so it binds in the active site, that it's irreversible. So we call this a suicide inhibitor. So it binds so strongly to the enzyme active site that the normal DUMP cannot bind there, and so this enzyme is effectively shut down, cannot produce TMP, and then cannot produce, again, the building blocks to undergo DNA synthesis. So again, that is one way that a certain chemotherapy agent works. So this is called fluoro-U. So fluorouracil is a very potent chemotherapy agent. So again, you can directly interfere with DNA synthesis in that regard. Or as we're going to see down here, you can actually interfere with the production of the necessary cofactor. So remember we said DHFR. DHFR is the enzyme that's responsible for taking sort of the demethylated DHF and making it back into the methyl transfer agent THF. Okay, so THF will go to DHF when it transfers its methyl group to U to make it into T, and to get back its methyl group, it uses DHFR, dihydrofolate reductase. Okay, so here are several potent chemotherapy agents that interfere with the activity of DHFR. So we're not directly inhibiting thymidylate synthase, but we are inhibiting the ability to make the precursor, THF, and then this methylene THF that you actually need to do the chemistry. Last little piece to kind of highlight here is talking about nucleotide degradation. Okay, so again, we said that there's no problem really degrading pyrimidine nucleotides. That's our C's, T's, and U's. You basically snip off the phosphate, and that can be degraded fairly easily in glycolysis, and then citric acid cycle, ETOP. And then the base is pretty easily broken down to malonyl-CoA, which again can be used for the citric acid cycle, or, again, not too much is produced, but again can be responsible for fatty acid synthesis. The problem comes with the degradation of purine nucleotides. So purine nucleotides, again, once you snip off that sugar, you're going to degrade that purine base to uric acid. And uric acid is especially problematic because if you have elevated concentrations in the blood, if you are unable to excrete it in your urine. So again, I'm going to, even though this is the same information that's over here, I'm going to kind of use this figure at the right here to kind of explain this. So both A and G are broken down to a common intermediate called xanthine. So just like we talked about the synthesis of A and G went through a common intermediate that we called inosine monophosphate, the breakdown of A and G as bases are broken down into a common intermediate that we call xanthine. So xanthine is further broken down and modified to uric acid, and under normal situations, people will excrete this in their urine, no problem. Okay, people who have an inability to excrete uric acid in their urine will again have a buildup of uric acid. So buildup of uric acid in your blood, your body looks for a way to kind of get rid of this. And so what often happens is it will deposit these uric acid crystals in your joints, and this leads to gout. Not going to spend too much time sort of talking about it here, but we also talked about the ability of uric acid to play a role in fructose metabolism. That was part of your fructose metabolism case study that we had at the beginning of the semester. So just briefly mentioning, well, how do drugs work then that help patients with gout? Well, some of these drugs work by inhibiting the pathway that would convert xanthine into uric acid. So again, by inhibiting this pathway, you don't accumulate uric acid, but what you do accumulate is xanthine. Now, what your body does have is the ability to take xanthine and convert it into a different metabolite, which usually does not have the problems with excretion that uric acid does. So again, shutting off the pathway at this point just allows us to sort of siphon xanthine into another metabolic excretion pathway. So real brief, just kind of chapter here, a couple of disease application states, just talking about nucleic acids and nucleotides.